Now, as we turn to consider how protein molecules associate to form larger assemblies, it's illuminating to consider hemoglobin, the protein in the blood which transports oxygen from the lungs to the tissues. This is a multimeric protein made up of subunits of two different kinds. Here we see one of the alpha subunits in a space-filling form, which associates with one of the beta subunits, shown here in yellow, to form a quite strongly associated alpha-beta pair, related by an approximate twofold axis of symmetry. This pair associates with another identical pair to form the tetrameric molecule, which you see here. This molecule has quite high symmetry. There are two approximate axes of symmetry relating alpha and beta subunits, and there is a real axis of symmetry shown at right angles to this diagram, which relates the two alpha subunits and the two beta subunits. Let's look now in a little more detail at the contact between the alpha and beta subunits making up this alpha-beta pair. If we turn the molecule first, we can look edge on at the contact region between the alpha and beta subunits. And if we now take away the space-filling representation, we can see underneath the skeleton of the polypeptide chain, which shows that the chain in this molecule is folded very largely in alpha helical regions. If we concentrate on the white and yellow subunits, notice that the regions that come together are related by pseudo-twofold symmetry at similar regions, and the reason why they come together is that the subunits here have sticky patches made up largely of non-polar hydrophobic residues. The principles involved are just the same as those involved in the folding of the lysozyme chain, which led to a hydrophobic pocket in the middle of the molecule. Here, there is a hydrophobic pocket between two subunits. In addition to these non-polar contacts, there are also polar contacts involved, in which hydrogen bonds are formed between the nitrogen atoms, colored blue, and the red oxygen atoms. But these are buried within a very largely hydrophobic region. So they form a strong contribution in which the partial electrical charges are compensated. Overall, the impression is of a mainly non-polar contact in which more than 125 atoms are involved. And this association is quite a strong one. This alpha subunit and beta subunit remain connected in this way throughout the activity of the hemoglobin molecule. Hydrophobic cores and sticky patches then are very important in aggregation of protein molecules. Let's look now at what happens if we turn to an abnormal hemoglobin which undergoes a further level of aggregation. Hemoglobin is involved in carrying oxygen in the red blood cells, and the red blood cells shown here have this nice characteristic disc-like shape, a biconcave disc. In sickle cell anemia, however, the blood cells are sometimes distorted into these grotesque sickle shapes. And if we disrupt one of these sickle cells, we find within it a mass of fibers formed, rather than a concentrated solution of hemoglobin. The next stage in our investigation of the aggregation of protein molecules, then, is to consider how it is that hemoglobin molecules can come together to form these fibers in sickle cell hemoglobin. If we look at normal hemoglobin, the tetrameric molecule, in a space-filling representation, we see that the surface, as usual, is largely polar in character, with nitrogen and oxygen atoms exposed upon it, but together with a number of non-polar residues here and there. If we take away the space-filling atoms, apart from one or two, to expose the skeleton, we can concentrate on this residue, which is a glutamic acid residue, number 6 in the beta chain. In sickle cell hemoglobin, this polar residue is changed to a valine residue. That's a change to a totally non-polar residue exposed, as we see, on the surface of the molecule. Elsewhere, on this right-hand side of the tetramer, there is another pocket down here, which, despite the appearance of some oxygen atoms in it, is largely non-polar, made up of a phenylalanine and a leucine side chain, which are exposed only in the deoxygenated form of hemoglobin. That's hemoglobin which is not carrying oxygen. In other places on the molecule, there are also some small sticky patches, in particular in this view at top and bottom, where we see them again including some polar atoms, but largely non-polar in character. But these sticky patches are not very extensive. They're not large enough to lead normally to the aggregation of hemoglobin. But they are large enough to lead to the transient formation of aggregates involving 2 or more hemoglobin molecules, and we can see such a dimer now in which 2 of these sticky patches, the top of one molecule and the bottom of the other, have come together. But this is a rather weak interaction. It wouldn't persist for very long or lead to the formation of even larger aggregates unless it were reinforced in some way. Now, in sickle cell hemoglobin, we can imagine another such pair of molecules approaching our first pair in such a way that the new valine side chain forms a contact with this pocket which involved phenylalanine and leucine. And similarly, the phenylalanine-leucine pocket on our new hemoglobin forms a contact with the valine on the bottom hemoglobin of our first pair. Now this means that this molecule is linking together these 2 molecules and reinforcing the effect of this rather weak sticky patch, enough to make this a rather permanent aggregate, which, as we see, links together these 4 molecules. And since this contact is exposed now down here, and this contact is exposed up here, this is an open-ended form of aggregation, so that if we look at this tetramer and then how it might build up into a polymer in a slightly less well-resolved picture, we can see that long chains of hemoglobin molecules might form. We replace these by red representations for each tetrameric molecule. We now have a long fiber with helices of hemoglobin molecules intertwined. That seems to be the way in which sickle cell hemoglobin molecules aggregate to form the fundamental fibers. And if we go back to the electron micrograph showing one of these fibers in detail, we can then begin to see whether this structure is observed. We home in on this picture, looking at it at slightly larger magnification, then we can begin to see regions such as that one, where one can just about discern the separate hemoglobin molecules forming this long, twisted helical array. So in this way, we can understand how it is that a simple change in the hemoglobin molecule, a change of 1 amino acid residue in each of the beta subunits, can lead to the formation of long fibrous arrays of hemoglobin molecules that distort the shape of the red blood cell into the shape that's characteristic of sickle cell anemia. And what's more, we can go on and try to intervene in this molecular situation in such a way as to ameliorate its pathological effects.